Welcome everybody to today's QBI SBI seminar. It's a great pleasure to have Dr. Balyn Zaro as a speaker here. Balyn is a member of the Helen Diller Family Comprehensive Cancer Center and the QBI Institute. She uses very interesting chemical biology tools and mass spectrometry to look at innate immunity, also hematopoiesis, and some other aspects of human diseases. So that's quite nice and interesting approaches. She graduated from the University of Southern California, and then also did postdoctoral studies at the Scripps Institute with Ben Cravert, and joined the faculty in UCSF in 2019. So she's one of the more recent additions. Great pleasure to have you here with us, and I'll hand over to you, Balyn, for your talk.
Thank you so much. It's a pleasure to be here from afar. I'm going to share my screen and hopefully you can see. All right.
Yeah, looks fine.
Everything good? Okay. All right. So my talk today is entitled "A Chemical Biology Approach to Studying Innate Immunity." And this is a real survey of what my lab is currently working on. So there's a combination of published and unpublished work that I'll be sharing today. Before we go too far, I just think I wanted to lead with acknowledgements. Starting a lab in a global pandemic comes with all sorts of challenges. And these are the brave individuals, Reagan, Jose, Sarah, Kate, and Andy, who joined my lab and have really produced all the work that I'm going to show today. And I also want to take a moment to acknowledge our really generous collaborators, in particular Irv Weissman, Mickey Tall, and Jennifer Coburn, who have really enabled us to do what I think you'll see is a really broad range of types of experiments, despite being a small lab, navigating sort of biosafety protocol setup and the confines of starting a lab in the pandemic. And of course, our funding sources who've been really generous in supporting this work. So since this is a broader interest talk, I wanted to make sure that we are all on the same page with what the innate immune system is. And so this is a slide that was taken from biorender.com that was made by Akiko Iwasaki at Yale, that I've sort of adapted a little bit, which kind of breaks down simplistically the difference between the innate immune system and the adaptive immune system. And we really think of the innate immune system as a quick and non-specific response to things that are found in our body. And this really is a surveillance mechanism that allows our immune system broadly to identify things that should or should not be there. This is something that we think of on a very short time scale, which allows then time for the adaptive immune system to come in. So the innate immune system that you are born with is what you'll have throughout your entire life. And it's a much more complex system than the adaptive immune system. Despite this fact, relatively little is known about the innate immune system. So if you look at your immunology textbook, you may have like 9 chapters on the adaptive immune system, and the innate immune system will be like 1 chapter, and it usually starts with the innate immune system is less well understood than the adaptive. And so if this is totally overwhelming to you, if you're coming from more of a chemistry background, I just ask you to remember that the innate immune system has developed over millennia in response to generational pathogen exposure. So this is a germline encoded immune system that doesn't appreciably change over your life. So what are the benefits of studying the immune system? Well, I just described to you the fact that this is a genetically stable milieu of cells that's relatively unaltered during your lifetime. And so what that means is that there's potential broad application in sort of anything that you discover. So how an immune cell responds to a cancer cell or a pathogen may be much more similar than you would appreciate compared to an adaptive immune response, right? Where you have a very specific cell type that's identifying a very specific, cell, disease cell, for example. So I just told you why the innate immune system has so much promise. Here is the sad truth. If we look at currently approved innate immunotherapy, specifically in the cancer space, you will see that there is nothing, whereas the adaptive immunotherapy space is, rife with drugs that have really changed the way that we treat a lot of human diseases and the way we think about treating these diseases as well. And so my lab really broadly across, decades long, hopes to contribute to filling in this empty space. And when I show this, it's not supposed to be this tall order that my lab alone is working on. There have actually been several companies that have recently had multi-billion-dollar exits where they are only focused on innate immunotherapeutics. Companies such as Genentech have also multi-billion-dollar collaborations with companies that are developing innate immune-specific therapeutics. So there's a ton of interest in this space, and there are clinical candidates, but there is nothing that has been clinically approved as an innate immunotherapy. And so in order to kind of develop these therapeutics, we're really thinking about focusing on macrophages, which are the cornerstone of the innate immune system. And we like to simplify this by thinking about how macrophages surveil around our bodies to sense self and non-self. And to simplify this even further, we think about non-self interactions where a macrophage wants to find something that shouldn't be there and eat it in a process called phagocytosis. And this can be regulated in a non-self scenario where an eat-me ligand is expressed on the surface of a cell, and that can engage an eat-me receptor on a macrophage and allow the macrophage to eat. Conversely, in a self-recognition scenario, a macrophage has a don't eat me receptor that can engage a don't eat me ligand, and this physically blocks the ability of the macrophage to cytoskeletally rearrange and eat the target cell. And so I'm going to tell you two stories today. The first story is a self don't eat me story, and the second story will be a non-self eat me story. So the project that I'm going to be talking about in my lab is led by Sarah Warrington and Reagan Volk, as well as our collaborators, Mickey Tall at MIT. So the most widely studied don't eat me signal exists between the don't eat me ligand on a mammalian cell called CD47 and the don't eat me receptor on macrophages called SIRP-alpha. And this ligand receptor interaction is implicated across basic and disease biology. So likely if you're studying a biological process, CD47 SIRP-alpha may be involved. And what I mean by that is like CD47 is important in blood formation, it's important in cancer, infectious disease, and synaptic pruning in the brain, in neurodegenerative disease, in cancer, you name it, it is likely involved. And so this has made it an attractive therapeutic target in the context of human diseases, but also because of its sort of broad-ranging existence, has also made it a very complicated therapeutic target as well. So a paralog of SIRP-alpha is called SIRP-beta, and unlike SIRP-alpha, SIRP-beta does not bind CD47. And this makes sense in the context of SIRP-alpha signaling as a don't eat me receptor that has an intracellular inhibitory motif that blocks phagocytosis from occurring. Conversely, SIRP-beta dimerizes with DAP12, which is a pro-phagocytic or eat me type signal. And so it makes sense that CD47 wouldn't bind. And structural studies that were conducted by the Barclay group at Oxford have demonstrated that there's a single amino acid that if you mutate in the CD47 binding region of SIRP-alpha or in SIRP-beta can actually allow CD47 to bind. So this is really surprising that SIRP-alpha and what's really surprising about SIRP-alpha and SIRP-beta is that these two proteins in the extracellular domain have over 90% sequence homology. So this is a really specific interaction that is kind of uniquely engineered where SIRP-beta cannot bind, but SIRP-alpha can bind 47. And to date, there's no known ligand of SIRP-beta. So we were interested in understanding the human genetics of SIRP-alpha, and if you look on Nomad, what you'll notice is that statistically across the entire gene that encodes SIRP-alpha, there's not a ton of variation. But if you look more closely at the exon that encodes the CD47 binding domain of SIRP-alpha, which is here, there's actually a ton of variation. And shown in this table are variants that have been detected across sequence individuals in at least 10% of the population. And you can see that there's a ton of missense variants as well as some frame shifts and deletions. And this is actually a ton of variation in a relatively small sequence. And what we really were wondering is why? Why would this happen? It doesn't really make sense. This is a self-recognition event. It's not as if our SIRP-alphas on our macrophages are constantly seeing other individual CD47. CD47 is also highly conserved across the population. So again, it doesn't really make sense. However, SIRPα is an innate immune receptor. And as I told you at the beginning of this talk, SIRPα, or the innate immune system, has been developed over millennia in response to generational pathogen exposure. And so this led us to ask the question, is all of this variation a cause or a result of selective pressure by pathogens? And in order for this to be the case, right, we would need to have a pathogenic protein that can bind to SIRPα. And so we started thinking about what sorts of pathogens would do this, right? And not only what sorts of pathogens would do this, but what's actually easy to study. So we wanted to find extracellular pathogens that we knew were difficult to clear, but were relatively easy to obtain and culture in a laboratory environment, and were common enough potentially to give rise to some degree of polymorphisms. Not all of them, but maybe we're contributing to this. And one of the pathogens that we looked at is Borrelia burgdorferi, which is a bacterial spirochete that's responsible for the pathogenesis of Lyme disease. And so shown here is just a GFP positive Borrelia, where you can see that it's kind of this like squiggly long pathogen. And we stain this with either CD47 affinity reagent or isotype control. And what you can see is that with our CD47 affinity reagent, we saw really significant staining across the entire spirochete. And we've quantified the binding over isotype control for both the human CD47 affinity reagent as well as a mouse. The humans specifically seem to have some sort of affinity on the surface of these Borrelia that we were really excited to understand further. So we decided to first identify what this protein was on the surface of Borrelia that could be binding. And so through an IP mass spec experiment and a series of filters, we identified this protein as this integral outer membrane protein P66. It's not a very exciting name, and it's a 66 kilodalton protein, but it's been shown to be required for infection persistence, but not for establishment of infection. And like CD47, P66 is a integrin binding protein. So the first thing we did was blast search this against CD47 or align it with CD47, and it doesn't show up in a blast or in an alignment at all. So this suggested to us this was a mimic in structure as opposed to sequence. We took recombinantly expressed P66 and demonstrated that not only could it bind to our CD47 affinity reagent above isotype control, but that it could also bind to a SIRPα Fc. We wanted to go further and to quantify the affinity of this interaction between P66 and SIRPα and compare that to CD47 and SIRPα. And what we were really surprised to find is that SIRPα has a higher affinity, almost 50-fold higher affinity for P66 compared to CD47. And we next wondered, given the high sequence homology between the CD47 binding domain on SIRPα and SIRPβ, is it possible that P66 binds both receptors? And in fact, we were able to demonstrate that P66 binds to both SIRPα and SIRPβ with comparable affinity between these two interactions. And this represents the first known ligand of SIRPβ. And this is really exciting because now we can start thinking about understanding more deeply the biology of SIRPβ. So now that we had identified this CD47-like molecule that could bind both SIRPα as well as SIRPβ, we wanted to ask the question of who would win in a head-to-head competition, right? Is P66 signaling as a don't eat me signal or an eat me signal potentially? And so to do this, we took primary human monocyte-derived macrophages and we challenged them with either wild type or P66 deficient Borrelia that we obtained from Jennifer Coburn. And before we challenged the macrophages with these bacteria, we pre-treated them with a pH-sensitive rhodamine dye. And so what this allows us to do is that when this dye enters the acidic lysosome of the macrophage, it fluoresces. And so over a course of several days, we can actually image phagocytosis occurring. And so the number of events is the number of phagocytic fluorescent events that we're seeing on the microscope. And what you can see right away in blue is that P66 deficient Borrelia is eaten faster and better by macrophages in an in vitro binding or in vitro phagocytosis assay. And this suggested to us that P66 is really functioning as a don't eat me signal. We've been doing additional studies, and preliminary work suggests that this is likely due to the fact that SIRP alpha is a much more dominant protein on the surface of macrophages compared to SIRP beta. And we're really interested in understanding this interplay between SIRP alpha and SIRP beta and P66, and whether or not this is really this dominance of the don't-eat-me signal is being driven by SIRP alpha abundance. So digging more deeply into the importance of P66 in the context of Lyme disease, since this was found on this Borrelia pathogen, we looked at data that was previously published by the Green Lab that looked at the antigenicity of different proteins on the surface of Borrelia and how they correlate with the symptoms that an infected individual has. And so what we saw is that individuals who generate more antibodies against P66 have less severe symptoms. So there's an inverse correlation between symptom severity and P66 antibody generation. We also have preliminary data in a return to health versus a persistent Borrelia infection model that suggests that individuals who are able to return to health following a course of antibiotics it also generate higher antibody titers against P66 compared to individuals who don't return to health. And what's important to note in this context is the fact that these patients all had tick bites. They all had the canonical bullseye rash. And so the only difference is that after the course of antibiotics, they didn't return to health. And actually you're able to culture Borrelia out of these infected individuals who didn't essentially respond fully to the antibiotics. And so this has kind of led us to ask this question of, are these subsets of individuals who don't return to health or have more severe symptoms, is this potentially related to SIRP alpha polymorphisms? And could this be a key indicator for return to health following Lyme disease diagnosis? And to dig more deeply into this, we started looking at individual donors. So shown here are 4 representative donors from that phagocytosis assay I showed you. And so this is with wild type and P66 deficient Borrelia. And what you'll notice is that all of the donors eat P66 deficient Borrelia better, but there are very different rates of phagocytosis across the board between each donor, and also sort of their response to a P66 deficient Borrelia versus a wild type. And so we're really now going back to think about looking at these different donors and looking at their SIRP types and asking if SIRP polymorphisms are important in understanding how this infection can be cleared. As additional evidence to the importance of SIRP alpha in human disease, it's been recently shown that bone marrow transplantation is more successful if you SIRP match your donor to recipient compared to if you don't. So it seems like SIRP alpha and self-recognition is highly varied across the population, and that this could be really important in how we think about treating diseases. So in this first half of my talk, I've told you about a bacterial mimic of a mammalian don't-eat-me signal. This is the first report of a bacterial protein signaling through a canonically mammalian signaling axis. We also have now found the first known ligand for SIRP beta, so we can really go after that biology and think about sort of looking at how the structure of SIRP alpha bound to P66 as well as SIRP beta bound to P66. We don't think that the global tick burden across evolutionary timescale is so great that it's been driving these polymorphisms. And so we think that there are other bacterial proteins that likely bind to both SIRP alpha and SIRP beta and may be engaging in some sort of host pathogen coevolution type scenario. These SIRP family polymorphisms may have broad-reaching implications in human health and disease. And we think this is really important. For example, there are 3 dominant variants of SIRP alpha, and they correlate or they're associated with different geographic locations. And so individuals who are, the dominant SIRP variant in East Asian cultures and East Asian societies is one variant compared to the one that's associated with those of European descent. So it's not too much of a stretch to think that perhaps, from an evolutionary timescale, that individuals in different locations have had selective pressure on SIRPs with different proteins. And so we think that this work is really challenging the assumption, one, that all humans accomplish self-recognition in the same manner, but that also this is distinct from how we engage pathogens, because with these levels of SERP variation, are we also seeing variation in the ability of an individual to bind to CD47, for example, as well as to bind to these bacterial mimics. So I'm going to shift gears abruptly to talk about another side of the lab, this non-self or eat me signal recognition that is being driven by Jose, a really talented third-year graduate student in my lab, and a technician, Andy, who just joined us in May. And they're really interested in these eat me signals. There's some level of complexity to this, and I realize this is kind of an overwhelming slide, but I will walk you through this. In order for a macrophage to eat, you need to have an eat me signal, but you also either need to block a don't eat me signal, or there can't be an eat me signal or a don't eat me signal. And so what this means visually is that if you have a don't eat me ligand and an eat me ligand and a don't eat me receptor and an eat me receptor, you will not get phagocytosis. These don't eat me receptors dominate the phenotype. If you block your don't eat me signal and you have an eat me signal, you will get phagocytosis. If you block your don't eat me signal and you don't have an eat me signal, you will not get phagocytosis. So it's very important, if we're going to be don't eat me biologists, to also be eat me biologists, so that we can understand the interplay between these two scenarios. And one of these eat me signals that's been implicated as an opposing eat me signal to CD47 is surface calreticulin. And as a mass spectrometrist, this was sort of surprising when I first discovered this, because calreticulin is a highly abundant ER resident chaperone protein, and it's always found in mass spec data sets. And I always look at it and ignore it. But in this case, we couldn't. So mammalian cells will often have surface calreticulin that binds to LRP, a receptor on macrophages, and this will promote eating. So if you knock out calreticulin, as shown in this paper, that you actually get less eating, even in a sort of apoptotic event. If you block surface calreticulin with a blocking antibody, you also lose the ability of phagocytosis to occur. So this is a pretty important eat me signal, but this really was surprising in the fact that I just told you calreticulin is normally not on the surface. And so the question that we had was, how does calreticulin make its way to the cell surface and function as an eat me signal? And it's been shown by Feng et al. In 2015 that BTK, which is Bruton tyrosine kinase, is implicated in the ability of macrophages to phagocytose and present calreticulin on their cell surface. And so what Mingye showed was that if you activate macrophages in any type of way with TLR4, TLR3, TLR7 agonists, that you can activate BTK, or you can activate to see phosphorylated BTK, and that this can be inhibited by a BTK inhibitor called ibrutinib. And importantly, this phospho-BTK correlated, or this activated BTK correlates with phagocytosis rates. So if you treat with imiquimod, which is a TLR7 activator, you can see increased phagocytosis over time. If you directly inhibit BTK with ibrutinib, you see a loss of phagocytosis really quickly. What's really interesting is that global calreticulin levels are unchanged, but if you look at cell surface calreticulin levels, imiquimod increases surface calreticulin, correlating with an increased phagocytic capacity and TLR7 activation, whereas ibrutinib, which inhibits BTK again, is going to decrease the amount of surface calreticulins available. And so this is really interesting, but to us as chemical biologists, we thought that potentially we needed, in order to really understand eat me signaling and the role of BTK in eat me signaling, we needed to have more direct and selective BTK inhibitor in order to validate this hypothesis, right? So imiquimod as a quote unquote BTK activator is actually a TLR7 agonist, and ibrutinib is a BTK inhibitor, but is actually not incredibly selective for BTK. So more specifically, ibrutinib is a covalent kinase inhibitor that irreversibly binds its targets. And so shown here is BTK bound, or ibrutinib bound to BTK at cysteine 481 here. And what you can see is that it perfectly fits in this one site, the active site of BTK, and works really, really well. This is kind of the probably the most successful example currently of a covalent kinase inhibitor in the clinic. Was granted breakthrough status by the FDA in about 2014, and has gone on to, treat a ton of individuals. The idea being that if you can treat with this covalent inhibitor, you will irreversibly bind your protein, and that will result in not only like decreased dosage and dosage frequency, but also it was initially thought that this could potentially allow or minimize treatment-associated resistance mutations. This has subsequently been shown to not necessarily be the case, but this is what was originally thought to occur in the earlier days of ibrutinib treatment. So ibrutinib has totally cured a ton of people of blood cancers. But a lot of these individuals end up developing heart problems, and in particular atrial fibrillation, which then goes on to cause a slew of other problems for them long term. When I talk to cardiology friends, they'll say that essentially like ibrutinib can be that it's entirely new field of cardiology called cardiac oncology. It can basically be attributed to the rise of ibrutinib, where now all of a sudden you had all these cancer patients who were also experiencing heart issues. And the real challenge is that ibrutinib has both kinase and off-kinase targets, and particularly in cardiomyocytes, ibrutinib binds to the kinase CSK. And this off-target reactivity can be attributed to a time-dependent reactivity event. So initially, our inhibitor ibrutinib has really selective on-target engagement, but over time, it begins to engage with less favorable targets that ultimately results in a loss of selectivity over time. So there's been a ton of efforts to try to fix this. The Taunton lab here at UCSF developed reversible covalent inhibitors, the idea being that these inhibitors will have high selectivity due to their scaffold for their intended targets, and that the covalent interaction will have a longer residency time on these true targets compared to off targets that will reverse Michael out of the cysteine engagement. During my time in the Cravath lab, I developed metabolically labile covalent inhibitors, where they essentially creating time bombs on the electrophile of these molecules. So that way they are metabolically inactivated over time before they can engage off targets. And really, really intriguingly was this idea of using a tempered reactivity electrophile. And actually this resulted in a molecule that's clinically approved and widely used called acalabrutinib. And so what's really unique about acalabrutinib is that this has a less reactive warhead, a butylamide, that can still engage cysteine 481 on ibrutinib. And what's unique about cysteine 481 is that it has the pKa of the cysteine is decreased, which allows for increased this increased nucleophilicity allows for engagement of a weaker electrophile. And so it's thought that by sort of tempering the reactivity of the molecule, that you would only see engagement of truly reactive, like really, really reactive cysteines, and a lot of the off targets of ibrutinib have less reactive cysteines compared to cysteine 481. And I've, when in talking to this sort of cardiac oncology group, it's actually it was really interesting. It was told to me that acalabrutinib is actually more prescribed now, especially for individuals who live in more rural communities and maybe don't have access to like a really wonderful heart clinic because of the known off targets of ibrutinib. And so this is kind of, to me, as a chemist or a retired chemist and a chemical biologist, it seemed to me kind of sad and silly that we would be using a less effective inhibitor simply because of the side effects were so much more reduced with this molecule. And so as we were looking at this, Jose in my lab had this really astute observation, which is that BTK, in addition to having this unique reactive cysteine, also had this massive binding pocket right here. And what we had also noticed is that in the literature that people were able to elaborate an ibrutinib-like scaffold pretty like readily without a lot of issue. And that seemed really surprising considering that, a lot of other kinases, when you do SAR, an immediate drop off in affinity. And so we wondered if potentially we could just reduce off-target engagement via steric exclusion. So could we elaborate the electrophile enough to where it would prevent it from actually binding to other molecules? So we generated a fumarate library of ibrutinib molecules where we had a methyl fumarate, a T-butyl fumarate, and then this phenyl fumarate. And our goal was to not compromise on the reactivity of the electrophile, but still have improved selectivity, right? So basically the selectivity of acalabrutinib with the reactivity of ibrutinib. And so we first performed this cysteine reactivity assay to just show that our T-butyl fumarate had similar reactivity. This blue compound had similar reactivity to ibrutinib in terms of a cysteine reactivity profile. And the other fumarates were much more reactive. We went on to measure the K-inact over Ki of our compound, this ibrutinib T-butyl that we were most excited about it, compared it to ibrutinib as well as acalabrutinib. And while it had a lower K-inact over Ki compared to the ibrutinib, it had a comparable K-inact over Ki with respect to acalabrutinib. And so this was encouraging enough for us to continue forward. So as a chemical biologist, we wanted to take advantage of bioorthogonal chemical reporting in order to characterize the selectivity of our molecule. So this slide was generated before Carolyn Bertozzi won the Nobel Prize a few weeks ago. So maybe the people on this call were less familiar then. And for those of you who've been living under a rock, I'll just briefly go through this, where we can take mice, we can take cells, we can take lysates, and we can react them with a chemical probe that maybe looks like a small molecule drug, a lipid, a carbohydrate, an amino acid, anything that we want. But it is equipped with a biologically inert chemical handle that allows us to then tag our proteins of interest in this case, although there has been used across all sorts of biomolecules. Once we have this alkyne installed on our proteins of interest, we can then react it with the corresponding azid tag, and this could be an enrichment or an affinity tag or a visualization tag. And this allows us to ask questions about what does global labeling with our probe of interest look like, and then also who are the proteins that are involved in this. So we synthesized alkyne analogs of our ibrutinib probes, and we treated Mino cells, which are a B cell cancer that ibrutinib is used to treat. And we treated for 1 hour to 24 hours with each probe. And so what you can see is that at 1 hour, we already see really strong engagement of BTK. Compound 5 is the ibrutinib alkyne probe. Compound 7 is our T-butyl. So you can see that it's a little bit of slower of an engagement event. But if we move over to 4 hours, what you can see is that our T-butyl compound 7, is engaging BTK at comparable levels to that of the ibrutinib alkyne. And if we move to longer time points, such as 24 hours, what you can see is that where our ibrutinib alkyne probe has a bunch of other protein targets, our T-butyl alkyne probe seems to only hit one. So this was really exciting, but we wanted to identify what these proteins were. And so to do that, we used chemical proteomics, where we took isotopically heavy cells where they had heavy lysines and arginines, or light lysines and arginines, and we can treat these cells with either a probe, so our heavy cells are treated with a probe, or a DMSO vehicle. We can then lyse each of these cell populations, and we can combine our heavy to light lysates at a 1-to-1 ratio. We subject this combined lysate to copper catalyzed azid alkyne cycloaddition with a biotin azid and enrich for our biotinylated proteins. We then performed on-beat trypsin digestion and LCMS to identify the proteins that were enriched with our biotinylation, which are putatively all probe modified proteins. We could then use mass spec to generate heavy over light ratios. So this would be proteins that are selectively enriched when the cells have been treated with a probe. And anything that had at least a ratio of 2, we considered to be a quote unquote true target of our protein. And what we were shocked to find was that whereas our ibrutinib alkyne probe had 247 targets, our new compound, this T-butyl alkyne probe, had only 7. And all 7 of these targets were also targets of the ibrutinib alkyne. So for mass spec aficionados on this call, what you'll see on these volcano plots is that the ibrutinib alkyne has a ton of protein targets, and actually BTK isn't even that well enriched. Whereas with our T-butyl compound, BTK seems to basically be the only like extremely significant and highly enriched target in our no probe experiments. So I mentioned that time-dependent reactivity is a huge challenge in covalent kinase inhibitor development. And so we wanted to perform time course experiments to bin our targets by other time-dependent targets. So these are targets that are not going to initially happen in a 1-hour timescale, but are beginning to engage proteins at a 24-hour time point, for example, versus targets that are engaged at 1 hour and this engagement persists to 24 hours. And so we use the same SILAC format, but instead of doing a no probe experiment, we did a 1-hour probe treatment versus 24-hour probe treatment. We did the same, lyse the cells, 1-to-1 ratios of heavy and light lysates, click with our biotin tag, enrich. And then what we're really looking for in the context of a time-dependent target is we have a light to heavy ratio of at least 2. And so that would become a time-dependent target, whereas a ratio of around 1 would be a time-independent target. And so when we bin these for our ibrutinib alkyne probe, we saw that about 60% of them are time-independent targets, and 40% are time-dependent targets. But when we looked at our 7 proteins that are probe-modified with our T-butyl, there is only 1 time-independent target, which is BTK. This is really exciting for us because when you think about doing a lot of biological experiments, being able to wash out a covalent drug in kind of a tractable system for a lot of our types of experiments that we do is really difficult. And so being able to add in a molecule that doesn't have a lot of off targets really cleans up the data for us a lot. So all of the reactivity experiments that I've shown you so far have been comparing each probe either with a DMSO vehicle or itself at a different time scale. So we wanted to perform head-to-head comparisons to make sure that our T-butyl compound engaged BTK at comparable levels. And so we did the same SILAC type experiment where we had heavy cells treated with our T-butyl and light cells treated with our ibrutinib alkyne. And we can do the same exact ratio experiments. And what we would expect is that our BTK should have about a ratio of 1. And we see that our ratio is nearly 1. It's actually a little bit more than 1 because it's actually slightly better engaged with the T-butyl, but well within the limits of reasonable experimental work. And so what you can see is that a lot of targets are preferentially enriched by the ibrutinib alkyne compared to the T-butyl alkyne probe. And if we look specifically at BTK, where we took the same cells from this experiment, from this probe versus probe experiment, and we looked at 1 hour versus 24 hours and did a biotin enrichment and looked for BTK binding or BTK biotinylation, what you can see is that our ibrutinib alkyne actually between 1 hour and 24 hours loses engagement of BTK, whereas our T-butyl alkyne has almost comparable, maybe slightly more engagement at 24 hours. And what we think that this sort of experiment summarizes is the fact that the ibrutinib alkyne is actually much more promiscuous in the proteome. And so what ends up happening is you add in your ibrutinib compound, it immediately engages a bunch of proteins, including like all of the cellular BTK, but it also engages other targets between 1 hour and 24 hours. So when more BTK is synthesized, more BTK cannot be labeled because there's no more reactive drug that's around for the cells to take up. Whereas with our T-butyl compound, we believe this molecule is literally sitting around to modify newly synthesized BTK, which is kind of a really exciting place to be in terms of developing a chemical tool to be able to interrogate biological processes. So these experiments up until now, we've been using our chemical probe. And so for some of you who are real expert chemists and chemical biologists, you'll say, well, what about the actual inhibitor? Does the actual inhibitor have similar reactivity properties? And so to do this, we to get at this question, we performed competition experiments where we took our heavy or light cells and we can pre-treat the cells with either our inhibitor or no inhibitor. And so this means that any true targets of the inhibitor will now be prevented from binding to our probe that has the alkyne. So what we're looking for in this case would be a loss of enrichment of a protein upon pre-treatment with the inhibitor. So for the purposes of our ratios, a non-competed target would have a ratio about 1, whereas a competed target would have a ratio of less than or equal to 0.5. And this would be a scenario where the inhibitor has blocked the ability of the probe to bind. This can be visualized on the left-hand side of this panel by in-gel fluorescence, where you can see that BTK engagement is competed with both the ibrutinib as well as our T-butyl analog. And you can really see here like the just global promiscuity between these two inhibitors. There are bands that the ibrutinib competes away that the T-butyl does not, and those are denoted as examples with the red asterisks. With our T-butyl probe 7, we also see competition with ibrutinib as well as the T-butyl inhibitor. And we quantified this doing the same exact experiments by mass spec. And what we saw was that whereas the T-butyl had only 3 competed targets itself using our ibrutinib probe, ibrutinib had 34, and there was a 10 protein overlap between both proteins. And so this suggested to us that a lot of the selectivity that we're seeing with our probe is also conferred to the inhibitor itself. Now, there's a ton of work that's been done to distinguish between ibrutinib-sensitive cancer cell lines and insensitive cancer cell lines. And this is often used interchangeably with BTK sensitivity and insensitivity. So we performed cell viability assays with either a quote-unquote ibrutinib insensitive cell line versus ibrutinib sensitive cell line. And what we saw was that our T-butyl compound had different cytotoxicity profiles compared to ibrutinib. And actually, there was no real correlation between BTK dependence in this case. And so we think what's really happening is that a lot of off-target toxicities are defining this quote unquote ibrutinib sensitivity in a lot of cell lines. And so we think a lot more work needs to be done if we're going to sort of stratify cancers by their sensitivity to ibrutinib as a surrogate for BTK sensitivity. So in summary, I've shown you that these fumarate-based electrophiles can be used to kind of vary the steric bulk and reactivity of the molecule. The selectivity of our BTK inhibitor was improved by simply swapping out this electrophile and sort of conferring additional selectivity by simply doing a little bit of chemistry. And this was independent of warhead reactivity and metabolic inactivation. These esters are also actually metabolically sensitive. So we think that in an in vivo setting, this will confer even more selectivity. We're interested in applying this warhead to other covalent inhibitors potentially, and identifying protein targets beyond kinases potentially that can accept a bulkier electrophile as a means by which to increase selectivity. But the real question that we now are currently asking in our lab is calreticulin surface expression actually a BTK-dependent event? So trying to bring it back to this macrophage self-non-self recognition, and is BTK really important in this non-self eat me signaling event? So with that, I want to thank you all for your time listening to this virtual seminar. I know we don't have as many virtual seminars anymore, so I'm really appreciative of everyone logging on. I'm happy to take any questions on this call, but also if you are someone who's less comfortable asking questions in this sort of format, I encourage you to send me an email at balynzaro@ucsf.edu. So with that, I'm happy to take questions.
Yeah, thank you very much for this really interesting talk. Please put your questions in the Q&A box. There is already one question there from Brianna Biebel, asking when SERP typing is done for bone marrow donations, is that just SERP alpha or also beta?
So for now, it's just SERP alpha. SERP beta, there's like a handful of publications on SERP beta. So I think now having a ligand to really interrogate the biology of SERP beta will be really helpful.
Mm-hmm. Actually, I have also a question regarding these CD47 in the SERPs. How is the expression regulated? Are those constitutively expressed, or is the expression modulated by infection or any cytokines?
That's a great question. So SERP expression is constitutively active, it seems. The CD47 expression is what really varies. So an example of this is red blood cells. So young, healthy red blood cells express CD47. As they age, they lose CD47 expression so that macrophages can clear them out. Cancer cells upregulate CD47. And infected cells, so cells that have been infected by pathogens, will also upregulate CD47.
And is there actually any agents which can change that expression yet? So
No, the way that people have been targeting this currently is to block CD47 therapeutically with an antibody.
Okay.
And what they're really playing to in that scenario is the fact that cancer cells also have higher levels of eat me signals.
Mm-hmm.
And so that allows the cancer cells to be eaten, whereas a red blood cell, for example, doesn't have all of these or as many eat me signals.
And does this system also play a role in degenerative diseases? Like you have too many eat me signals and causing degeneration?
So we know that eat me don't eat me signaling is involved in synaptic pruning. And it's postulated that CD47 is important in neurodegenerative disease as well.
Okay, I guess I have asked enough questions. There is another question in the Q&A by Nick Riley. Great talk, Balyn, with the hypothesis that IPT-BU ALK hanging around to react selectively with BTK. Is it worth doing flux experiments to measure actual amounts of IPT-BU ALK that are in the lysates over time and correlating with BTK expression? Or does the competition essay answer that question sufficiently?
That's a great question. We would love to do those experiments. We just simply didn't have access to metabolism instrumentation. Like we don't have a triple quad or something to be able to do these measurements really accurately. So we thought that the competition experiments, as well as the biotin enrichment experiments, specifically looking at BTK or biotin-elated BTK, really were sufficient in this case for now. But that's a great question. We would love to do that. And we think we have reason to believe at this point that if there is a lot of like carboxylesterase activity around that the TBL compound will become inactivated, which also plays into the role of like the complexity in like a mouse experiment or something as well. But what we like about this compound is that we can dose with a lot more because it's hitting less, which is nice and surprising considering the cysteine reactivity profile.
All right. Is there any more questions? Not seeing any more questions. So thanks again, Balyn, for this really fascinating talk. And thanks everybody for attending and listening. And yeah, until next time.
Thank you so much.
Thank you. Bye-bye.
Bye.